Okay, so welcome everybody to the Curly Horse Genetics presentation being presented by Curly Horse Advocates. This is part one of 3 presentations that are planned so far. We'll see how we do with it. We'll see how much everyone is excited about everything we learned today. And we're going to hear from 2 people today. The first person is Dr. Mitch Wilkinson. He is joining us from, where is it, Mitch? Is it Colorado?
Pagosa Springs, Colorado.
Yeah. So it's very kind of Dr. Mitch to grace us with his time and presence. And then later on, we will be hearing from Dr. Gus Cothran as well. And after both of their presentations, we're going to do a question and answer session. So Dr. Mitch's presentation will probably be about an hour. And then we might have like a 5-minute intermission just to let people get a snack, go use the bathroom. And then we'll come back for about half an hour with Dr. Gus. And then after that, there will be a question and answer session. So I wanted to tell everyone a little bit about Dr. Mitch before he gets started. We'll just give him a little introduction. Dr. Mitch is president of the Curly Mustang Association. He's also the chairman of the Sulphur Springs Mustang Registry Research Committee, vice chairman of the International Curly Horse Organization's Research Committee, and a published international author of articles on Mustangs and curly-coated horses. Although not a professional equine scientist, he has had a lifetime involvement with horses. His family's breeding and interest in horses has spanned 3 generations. With a master's degree in biology, oral biology, and cancer genetics, if you were wondering, from Baylor University in 1990, he can serve as a coordinator with registries and individuals needed for a scientific project's completion. Dr. Wilkinson has initiated many equine research projects over the last 20 years and is essential in disseminating scientific research results to the general horse owner. He has also been involved with grant writing and procurement of research funds for equine genetic inquiries. Dr. Wilkinson's background in human genetics and scientific research allow a basis of understanding for his participation in current equine scientific projects. And with that, Dr. Mitch, I will mute myself and let you take it away.
Thank you, Bree. Over the past 70 years of my life, I've taken many turns. But one of the most significant turns began with my introduction to curly horses, which was around 30 years ago. My love of horses began in early childhood, as Bree said, on our family ranch and continues to this day. I'm not an equine scientist, and although I have had postgraduate training in genetics, I do not consider myself a geneticist. My career has been in medical and dental public health, but like most of you, first and foremost, I'm a horse lover. My association with Dr. Cothran began close to 20 years ago. Over this time, I have been a pain in his side about curly horses and an advocate for these horses. Every one of us here owes Dr. Cothran a sincere thank you for all that he has done for our horses. And we should also thank the staff of the Equine Genetics Lab at Texas A&M. In my published papers and videos, I try to check all facts presented with the real experts, people like Dr. Cothran. I really do stand on the shoulders of giants. This extremely diverse group of horses we're talking about can only be preserved for future generations by knowing verifiable scientific facts about them to guide breeding and management decisions. For too long, these horses have been the subject of speculations, legends, total fantasies, which are not based on any empirical evidence whatsoever. This has led to rivalries, misunderstandings, and disputes about philosophies, which are totally based on fantastical speculations. And this was much to the detriment of these horses. We all need to remember, this is not about our egos, your egos, or my ego. It's about the horses. It's about the future generations and preserving something special for them. And this is a cause that is far greater than all of us. And having made my little beginning statement, I'm going to go into the slideshow now. Let me go back one other time. We're going to be using a new application for an old technique, which is called principal coordinate analysis. And this has all been done by Dr. Cawthron, the staff of Texas A&M University. And I wish to thank them so much for their efforts that they have put towards our horses. There are a great many different types of horses. Each type is extremely different. The types that I have mentioned here are Missouri Foxtrotter types, Demelli Mead domestic type horses, Northern Nevada Mustangs, Southern Wyomings, Sulphur Mustangs, miniature horses, and ponies. And each type is distinctly different and has a different background. So that's what we're looking at, a very diverse population of horses. And this is just North America. This is not including other parts of the world. Now, right here in this slide, I show the various mutations that are found in North America so far. One was only recently found 6 months ago. But the KRT25 mutation is the most numerous, and it's found in domestic and feral populations. This has not been found in populations outside of the continental, outside of continental North America. And we have tested the majority of horses in foreign lands. They do not have KRT25, nor do they have SP6. And SP6 is another mutation found in North American curly horses. It was probably a spontaneous mutation that happened in domestic horses in the 1950s. In all probability, that's how that happened. It has never been found in a feral horse population in North America. And then we have the Cook mutation, which is found in most domestic and feral populations. The sulfur mutation, which is found in the sulfur herd of southern Utah, and our recent addition, the unknown curly gene A. And I called it that in case there happens to be an unknown curly gene B out there somewhere. And this was hidden in plain sight in the domestic population since the beginning of the domestication of curlies, probably going all the way back to the Demelli breeding program. And how this popped up, it probably was in the feral population. But again, this is a new gene. We don't know a lot about it. And these are the many different genes that can be found in North American populations. Sadly, the Pendleton curly gene in Oregon on the Cayuse reservation there is believed to be extinct. I'm planning a trip to Oregon to look into that a little further, but as far as we know, it's extinct. And the interesting thing about this particular gene mutation that we don't have anymore is they didn't lose their curls in the summer, which is an interesting development. And next you'll see the curly gem line that is associated with the SP6 mutation. And that particular horse in the picture is homozygous, meaning he has two copies of that particular gene. I think that's Kansas Dream, I'm thinking, if I can remember correctly. And then the KRT25 mutation is found in domestic and feral horses. And that's my horse Lucky there. And he is heterozygous for the KRT25 gene, which means he has one copy. And then down below is my old friend Nipper, who is a representative of the Cook gene. And if we can ever isolate that gene before it goes extinct, because there's so few horses that have it now, we may be able to test Nipper that we have on file to see if he is homozygous or heterozygous. And then there is an odd curly type gene that is found in some Spanish Mustangs. And I'm going to go into that deeper. I'm not totally sure that that's truly a curly gene, but then who am I to decide? So we'll go into that question a little further. And then finally, at the lower right, we have Mescal, who is just a beautiful, beautiful example of a sulfur horse, and one of the very few sulfur horses that is born with the sulfur curly gene mutation, which is different than all the others out there. And we only have 1 or 2 horses left that carry this, and we're trying so desperately to keep this particular gene from going extinct because it's gives a beautiful coat. And the next slide here. Anybody who's breeding needs to understand about Mendelian inheritance. When you breed 2 animals together, the result is not a blending of the 2 animals. You either inherit a gene or you don't. And this is called a Punnett square, and there are various places where you can look these up on the internet, and they will do the program will do all the work for you. If you just Google in Punnett square, you can come up with this. And here's a Punnett square of the mating of 2 heterozygous individuals. The big A is probably the mutation that gives a trait, and the little a is the wild type that is like all the other members of whatever this is. And as you can see, in a mating of 2 heterozygous animals or plants or anything, you will get a homozygous, or 2 copies of the big A. You will have, and that's a 1/4 probability, 25%. And then you will have a 50% probability of having a heterozygous, which are the green squares. And that would have a copy of the wild type and a copy of the gene that gives whatever particular trait it is. And then you will have a 1/4 chance of having a individual born that doesn't have the trait at all. And our curlies follow the same scheme. If we bred 2 heterozygous curlies that each one had 1 copy of KRT25, it would end up with these same odds. And the Punnett squares get more complicated as more traits are involved. And yet there are programs that can help you with that. And you need to be aware of this if you're breeding animals, because if the animal comes up and does not have the trait, it doesn't have the trait. It's not a blending of the parents. So keep that in mind if you're breeding, and everybody should be familiar with that. And this is another real important slide here that applies to our curlies. In almost every gene mutation that causes curly coats that we've identified, we have found that it is not a complete dominant expression. It's a semi-dominant expression of that gene. And here's an example. If you look at the roses on the right, big R giving you a red rose and little r giving you a white rose, in complete dominance, as you can see, you cannot tell the difference between the homozygous, or having 2 copies of the red, and the heterozygous, or having 1 copy of the red. The phenotype, which is the appearance, looks the same in both. And yet in semi-dominance, as you see over here on the right, you have a red flower, you have a white flower, and the pink flower is different than either one of the parents. And that's how our particular horses are in this scheme of things. They go and have a semi-dominant mode of expression of a dominant gene. And there are a lot of words for this. Semi-dominant is synonymous with incomplete dominance, intermediate dominance, and And these are a mode of expression. Now they're passed on as a dominant gene, but they're expressed in a semi-dominant way. And here is a good example. This is KRT25. And here's my good buddy Lucky again. And he has one copy of KRT25. And the horse on the left has two copies of KRT25. And these are both the common phenotypes or appearances of these particular of the KRT25 causative gene. And at this point, I want to make very clear what the definition of extreme should be. The Curley definition of an extreme should be a homozygous KRT25 horse, or a horse that has two copies of KRT25. And what you're seeing on the lower left is the extreme phenotype. And this term has become a little too broad at this point. And so we need to distinguish that is exactly what extreme means. And you're going to understand why that's important later on in the slideshow here. Now, this is really important here too. You need to look at the type of horse and not the mutation. Not that the mutation is not totally, completely important, but you need to realize that any horse can inherit any mutation, okay, depending on what mutations the parents are carrying. So do all SP6 horses look like Missouri Foxtrotters? No. There are Demelli horses that, through the rules of Mendelian inheritance, inherit SP6, but for all intents and purposes, the majority of their breeding is Demelli. And do all KRT25 horses look like Demelli horses? No. There are some that could look like Missouri Foxtrotters, depending on the makeup of the parents. Now, this is interesting. Does having a curly gene give a horse a good temperament? No, simply because there are two reasons because of that. One, we have an awful lot of curly genes, and it would be just kind of, it would defy the odds of nature if just having one of the many curly genes gives you a temperament, because these are all different genes. And temperament is a very complex subject. It has to do with environment. It also has to do with inheritance. And one of the very first genes that tended to be associated with temperament is the DRD4 gene. And that has to do with dopamine receptors. And it's on the 12th chromosome of a horse. And it has to do with curiosity, which is being curious and not spooky about things, and vigilance, which is being very aware of your environment and very spooky about your surroundings. But this is only one facet of temperament, and there are probably many other genes that are involved, plus the environment. So temperament is still being explored, but I can assure you that simply having a curly coat is not one of the many curly coats, is not a precursor for having a good temperament. And then last of all, I say, do all curly horses have a common origin? Obviously not. They're all completely different types of horses with completely different types of genes. And now I'm going to talk about the most predominant horse, and we're going to take a look here at Benny Demelli's breeding program and Copper D. And Copper D had, this is one of the few pictures of Copper D. And when I look at this picture, he surely had KRT25. He possibly could have also been dual gene for the Cook gene. And his father was a quarter horse, so he was only like half curly. And that was the original one. We may never know exactly what genes copper D carried because that was so far back in the past. And again, maybe we'll be able to trace that back, but maybe not. And now this is a, excuse me, jumped a slide. This is a real important slide here. And I think this is really important here. Pat Doak took this excellent picture of this KRT25 curly Mustang that's in the middle. She takes excellent photographs. And here's my good friend Lucky. And you notice that they really are two different coat types. Lucky has, anybody who's been around Lucky, he has just this virulent, lush winter coat that gets to be over an inch thick. And this other one has more of a wavy coat, but they're both heterozygous for KRT25. And what could possibly be the difference? Well, the difference are other genes, okay? And I don't have a horse model for this, but I do have a dog model. And recently, the coat length in dogs is found in genes called fibroblast growth factors, and fibroblast growth factor number 5 is what makes the difference between a long-haired dog and a short-haired dog. And just like horses have KRT25 mutation to give them a curly outer coat of hair in the winter, dogs have a KRT71 mutation, which makes a poodle, which gives it a curly coat. But this is another interesting thing that is found in dogs. Recently, there's a cis regulatory enzyme on the dog's 28th chromosome that had a SNP in it, and it negated that which coded for a transcription factor, which allowed dogs to have a second inner coat in the winter, which is found on wolves. It's found on huskies. It's found on malamutes, but it's not found on dachshunds. And so that has been bred out of the short-haired dog population. So the difference between these two horses may be that Lucky has a very distinctive second inner coat, which is a downy, fluffy layer. And then the curly guard hairs grow out from that. Now, he sheds both coats during the summer. Now, if this horse in the middle did not inherit that middle coat layer, then he is going to have a shorter, more wavy type coat. Now, the little horse over here on the right is a Spanish Mustang that was born with what we call bunny fur in the curly world. And what we could be looking at here is a horse that was born without a curly outer coat gene. And we're actually looking at that inner second coat, that inner fluffy coat, because this little guy is negative for KRT25. And if dogs are a model of something like this, the same thing could be happening in horses. Now, get me correctly here. This is purely speculation on my part, but it does seem to fit that something like that might be occurring. And dogs are the only true model that we have. And now let's look at another horse in the Demelli breeding program. These were the 3 genes that the Demellis were breeding. They were breeding KRT25, they were breeding Cook, and they were breeding a recently discovered gene, unknown curly gene A. And the Demelli breeding program added Arabian, Morgan, Appaloosa, quarter horse, and several other types of horses to the local northern Nevada Mustangs, which today are only represented by curly Mustangs from Fish Creek. And let's look at Colonel Austin, who was a very famous breeding stallion that the Demellis had. Cotton Eye Joe, nobody knows where he came from. He was either a feral horse or he was the foal of a feral horse. The Demelli buckskin mare, an awful lot of balding seems to trace back to her. And Colonel Austin had them in his pedigree twice, both of those. And then he also had Nevada Red, which was an Arabian. So and when I look at Colonel Austin and I see this unusual tail morphology, can everybody see that? Where the top of the tail is kind of naked, and then you just kind of have this puff at the end. That's characteristic of unknown curly gene A. That makes me very suspicious. Now, we may never know what he carried, but there's a possibility that he had that gene. And now I'm going to look at the second most prominent type of curly horse in North America. And that's a line of curly gem and the SP6 mutation. And keep in mind, the SP6 mutation has never been found in a feral population. And curly gem showed up in the 1950s at an auction. Nobody knows where he came from. And to show you how things came down in 1954, I want you to look at that first line of mares that he was bred to. The vast majority of them are unknown. The others that are known and have a name are simply grade mares. We do have a saddlebred, which is the only one associated with a breed over here, but we don't even have a name for that saddlebred mare. We don't even know if she was registered or if she was full blood. And so their offspring, curly gem's offspring, all the way down the line, and on the blue on the far right, you will see that that's the line of Shetland ponies that descended from curly gem. And as you look down through that generation of offspring through here, you see several of them that you will notice are very famous. Ginger started a very famous line, and she was bred to Tennessee walking horses and Missouri foxtrotters. Keep in mind that Tennessee walking horse was part of the makeup of Missouri foxtrotters back when that breed was being created. And then another very famous, and Sir Patrick came from that line, and all his descendants. And then Blaze, which we've all heard the name, was bred to a Missouri foxtrotter that was not curly, Walker's Merry Lad, and that began Walker's Prince T, which was a very famous stallion in that line. And then Gentleman Jim over here on the right started the line of curly ponies and miniatures. And so I just want to emphasize to everybody the humble beginnings of the curly gem line. And from the generation of Blaze on, they were very heavily bred into Missouri foxtrotters. So much so that they have become very close to that. And we're going to explore that in a minute. But here, and I misspelled, or this slide misspelled Merry Lad. That's M-E-R-R-Y lad. And he was a straight Missouri foxtrotter. And look, Curly Jim went to Blaze, and the Bradford mare and Curly Jim begat Blaze. And we have no idea what the Bradford mare was. And then Blaze was the mother of Walker's Prince T. And that really is that next generation after Blaze is where that particular line seemed to go. And here's a picture of Walker's Prince T. And there were 3 of them, if I'm not mistaken. And let me go back. Wait a minute. There. And now, at A&M, we were using principal coordinate analysis. And this graphically displays on a 3D graph the genetic relationship of, in this case, horses. And if you look at this particular graph here, non-curly Missouri foxtrotters are represented by yellow squares circled in red, and curly curly Jim descended horses are in blue, and they're circled in blue. And just like on a Venn diagram, you notice that they overlap. And the horses that are within that overlap area are genetically very, very close and difficult to distinguish genetically one from another. So what this graph is telling us is that because of the extensive breeding into Missouri Foxtrotters over the years, the curly Jim line of curly horses are, number one, a separate population, but a population that is very closely related to the Missouri Foxtrotters. And there are some that are almost genetically indistinguishable from each other. But in this case here, if you look on the far right and the far left, the horses that fall in those areas are quite different. So they are two populations, distinct populations, but very closely related. And that's a real important thing to understand. And here we have an example. You have a heterozygous SP6 curly Jim descended horse on the left, and that horse is Old Boy, owned by Terry Smith. And then we have I think this is Kansas Dream, that is homozygous for SP6. And those are the two phenotypes or appearances of them. Now, there. Now this is real important. Back in 2016, Dr. Cawthron, prior to that, had isolated the KRT25 gene, but didn't realize that was the gene. And he rejected it because not all curly horses had that gene. That's back prior to 2016, we seem to think that all curly horses had the same gene. And then later, through a joint French American research effort headed by Dr. Cawthron, we were able to find that KRT25 was one of the main causative genes of curly coats. But then in 2016, as we were typing all the different horses and finding out that not all of them had KRT25, I came upon Chester, and Chester is owned by Angie Gaines. And I realized by a combination of genetic testing and looking at pedigree that Chester had to have had two genes at the same time. He inherited both KRT25 and SP6. And when I realized this, I was so ecstatic. I had to call everybody I knew and send emails out and call Gus, and it just went crazy about the whole thing. And everybody else was less enthusiastic about this than I was at the time. And later on in 2018, a German team isolated SP6, and we came back and we were able to test Chester, and he was heterozygous for KRT25 and heterozygous for SP6, and carried both at the same time. And through a very complicated Punnett square, you can see what the odds are with an offspring, depending on what mare he's bred to. But as further testing went along, we were able to find out this was not an unusual horse. This was fairly common. And this is telling me that it is possible to inherit multiple curly causative genes at the same time in the same individual. And this changes everything of what we know about curly horses. And you're going to recognize some of these guys, okay? They're pretty famous stallions. And these are all dual gene horses that have inherited a copy of KRT25 and SP6. And then here's another generation of horses that have inherited the same thing. And you may recognize some of these names on these horses. So this is a reality of our curly world that we're breeding with. Let me go back. Oop. Okay. And now I'm getting to the Cook mutation. And this is one of my favorite horses. I consider this horse my godchild. And this is Karma, owned by Christy Harvey, and a beautiful example of the Cook mutation. And this is interesting because her father was Nipper, who was almost all Cook, but her mother was from Demelli breeding and carried KRT25. But by the rules of Mendelian inheritance, it so happens that Karma only inherited the Cook gene. So physically, she is kind of half Demelli and half Cook, but she only carries the Cook gene. And as you can tell from Karma here, this is a beautiful curly coat, and there are very few horses left. We do not want to see this beautiful type of curly disappear from the earth forever. And I urge everybody to help in the conservation of this type of horse, because it is important to carry this on to future generations. And we haven't isolated this particular gene yet. Now, this slide here, because of the pedigree, we know that Karma can only be heterozygous for the Cook gene. But I'm looking over here and you see a picture of me with my old pal Nipper, and Nipper could have been homozygous, but we don't know because we can't test. We don't have a way of knowing that. And below that is Nipper's father, Houdini. Now, admittedly, Houdini is not a very attractive horse physically with his conformation and everything like that. But he did begat a whole line of Cook horses. And I'm looking at that phenotype. There's a very, very good chance that Houdini was homozygous and had 2 copies of the Cook gene. And so right there, we may be looking at what the homozygous phenotype is. And now I get to another favorite horse of mine. Okay, this is Renegade. Okay, and Renegade is a sulfur curly horse. And he has a unique gene, which is only found in sulfur Mustangs. And sulfur Mustangs live in Utah. And he's one of the last of his kind that carry this particular gene. And we are doing everything in our power to save this gene from extinction. And we don't know what this looks like when you have 2 copies. Now, I know from the pedigree that Renegade has 1 copy of the sulfur gene. I don't know, I have no idea what 2 copies looks like. So that's something important. In these sulfur Mustangs, and this is a non-curly sulfur Mustang, and it's sulfur Cheyenne. You can see a picture of me trying to get nuts. This is a different sulfur horse out of the mane. And these are some of the most attractive Mustangs out there. And they are very, very unique genetically. There's nothing even close to them. And here is Renegade's mother. And she is a classic example of a beautiful sulfur Mustang that carries the sulfur mutation, giving her a curly winter coat, and there's her summer coat. And these horses can have manes and tails going down to the ground. It's just a beautiful coat, beautiful conformation, beautiful horses. We shouldn't let them disappear. And this is an important principal coordinate analysis here, showing various types of curly horses. Look at number 11 down in the bottom here of the graph. That's sulfurs. Okay, they're not anything close or near to other curly horses. They're totally unique. And look over here on the left side, upper, number 1 and number 13. Those are South American curly horses. And we'll go into that again, and they are completely different. All the other curly horses somewhat fall into a little cluster there. And I'm going to go over each individual part of this graph because it's such an important graph. Okay, this is my horse and Blackfish. And Blackfish's father came from Fish Creek, which is probably some of the original Northern Nevada Mustang Curlies that the DeMellis took and altered. And his mother came from Rocky Hills, and he was born in Rocky Hills. And if you look at this graph, which is a blow up of the previous graph, and each point represents a thing called a centroid, which is, instead of having a huge cluster of individual horses and their data, we've consolidated it into kind of an average. And 2 and 12 are two clusters, and 2 is Simpson Park, Rocky Hills, and 12 is Fish Creek. And you see that they're a little bit different there. And they're both Northern Nevada Mustangs. They're in the same area, and yet they're a little different. And I got to thinking about it. And the reason that they're different is number 2, Simpson Park, Rocky Hills, is right next to the DeMelli Ranch. Remember the legend of when they turned Copper D loose into the wild herds? And there are probably other escaped DeMelli horses that bred with some of the wild horses. And it was enough that they changed them from the local Nevada Mustang, but they're still pretty close. And so I thought that was an interesting thing to look at with that. And now here, number 7 is the DeMelli breeding program. So this is telling me that the DeMellis didn't alter the existing Mustangs of Northern Nevada all that much. That you know, I have a phrase which says, you know what happens when you breed an Arabian with a Mustang? You just have a Mustang with more Arabian in it. Okay. And the DeMelli breeding program had so many local horses from the Northern Nevada Mustang group that they're adding to various other horses they were putting with them to upgrade them. They really didn't change them all that much. And here I have a picture of my deceased mare, Shelby, on the bottom, and she was a pure DeMelli breeding. And here's Blackfish up on top, which represents kind of the wild population. So that and as you see the conformation of those 2 horses, they're not all that different. You know, they have the shorter legs, the big hips, the low tail, the short back, the thick neck. They really didn't alter them that much. And I thought that was another interesting aspect. And here we go. This is a warrior line. And in the next episode here, I'm going to go into this a little deeper. But the warrior line was bred very extensively into, or quarter horses were bred very extensively into the bad warrior line, and by many of the beginning breeders of that particular line. And I thought it was kind of interesting. I was expecting these horses to be completely different, but they ended up being fairly close to the DeMellis. And we're still investigating this particular part of this. Now look at this. This is pretty interesting. These are the Wyoming Mustangs that have curly coats, found mostly around Assault Wells Creek HMA. And you notice that's a completely different type of horse, and our graph says so also. And this is the yellow arrow that you see up here. And these horses are thicker, heavier horses. They're physically have a bigger stature. They have longer backs. They have more muscling, and they are a completely different horse than a Northern Nevada Mustang. So I urge people that adopt these horses, let's try to keep this horse as it was, and try to breed it to other Mustangs, either curly or non-curly, from that southern Wyoming area around Salt Wells Creek. And I think that's something that everybody ought to be aware of, because we really don't need to be mixing these different types of horses up. And finally, this is to be expected, 3 and 9. Curly Jim and the miniature Curly Jim descendants are right close to each other. And down below here, we've got Sir Patrick and his line of horses and a cute little miniature foal up there that has a curly coat. And that's to be expected. Wait just a minute here. Good. And this is the part that Gus is going to cringe on a little bit, because here I'm kind of going out on a limb, and and this may be totally coincidental, but it ends up that the Cook horses, number 10, and the Mongolian curly horses and the Zabalskaya curly horses from southern Russia all fall within a little group. And notice 6 and 10 almost overlap. And I was expecting 10 to be over there by number 7, but instead it ends up near the Mongolian horses. And this is a picture of some curly Mongolian horses down below here. And we took the three groups of horses, the Zabalskaya horses in blue, the Mongolian in the white squares, and the Manipur ponies, because we have recently discovered that there are curly horses in Manipur, India. And we put them together, and we didn't have a lot of samples. I think maybe if we had more samples, we would see some overlap here, but they are genetically close to each other, which makes me wonder why the Cook horses are showing up there. Sorry, my mouse is touchy today. And here are examples of all of them here. There's a Mongolian curly, a Zabalskaya curly, and a Manipur curly. And all of these are not bred for curls. They just end up being part of a herd, and that gene is floating around in that population. And they don't particularly breed for a curly coat, and and it usually falls at about the 10% level of horses inheriting a curly coat, and that's about what we see in our American Mustang populations. And here's where I really go out on the limb. Okay, I've been searching the Thomas Dixon legend for at least 5 or 6 years. And I know his brothers and sisters. I know where he went on vacations. I know all about him, but I can't find a connection where he went to India and came back. It doesn't mean it isn't there. It just means I haven't found it yet. And all these horses that are somewhat related there tend to be along the old Silk Road routes that are shown there. And could it be that the Cook mutation could be some of the remnants of the horses that Thomas Dixon brought back to Eureka in the 1870s? I don't know. It's total speculation, but it makes me very curious. And I had to throw in Jet. He's a really famous Manipur curly polo pony, and he's very renowned in India. So they're familiar with curly horses in India. And here I'm hopping down to South America. And here I am in South America examining a curly horse in South America. And remember, these particular curly horses are not, look at the red arrow here, they are not related to any other curly horse. So it's not like a stray curly horse from North America made it down to Patagonia. This is an entirely different horse with an entirely different mutation. And here we did a principal coordinated analysis on this. And as you see here, the typical Argentine Carrillo horse is in yellow squares. And the curly Patagonian horses are in blue and white. And the difference is that the blue samples were taken in 1 year and the white ones were taken another year. And each one forms a distinctive population. So what that means is in this very isolated part of Argentina, that there is a type of horse that a certain percentage of their population has a curly coat from an unknown gene, and they are completely different than the other Argentine horses. And so this may be something that the world may want to help to preserve. And we're in the process of investigating the warrior lines and how close they are to quarter horses, and the pony lines to see how close they are to Shetland ponies. And I'm going to be publishing that in a paper that is based on this particular slideshow. And now as we get close to the end, I'm going to talk about the curly unknown gene A. And this was discovered because of a wonderful lady in Switzerland named Veronica Schmidt, who sent me an email one day saying, "What's wrong with my horse?" And it got me curious, and we got to looking into it. And the more I looked into it, I started compiling a family tree of that horse. I started thinking about inheritance through the Mendelian laws of inheritance. And I came up with unknown curly gene A. And again, I was so excited about this. I was just like, oh, wow, we got another curly gene. It was hidden in plain sight, and we didn't know about it. And again, I was totally enthusiastic. Everybody else, maybe not so much. But the point is that we have discovered it. And these are some of the phenotype characteristics or the appearance of this particular gene. During the wintertime, these horses have a fairly decent looking coat, as you see in that middle picture of the horse in the snow. The very same horse in the summer sheds most of that out. And again, we have that characteristic little puff tail at the end. And down below are pictures of a horse that has this particular gene, and we haven't isolated it yet. But as you notice, these horses tend to keep that unusual tail morphology through summer and winter. And that horse is named Salon down on the bottom, and she carries that particular gene. And look at this, here's Salon in summer and winter, okay? These horses get really sparse of hair in the summer, but they tend to keep that strange little tail. I don't know how else to describe it, but it's an unusual tail morphology on the coat. And that's the same horse summer and winter. And that's the manifestation of curly unknown gene A. Now, when we look at breeding on this, let's look at what we know and what we don't know. If we mix KRT25 and a sulfur, we have no idea what you'll get. You know, if we do it with a cook, we don't know. We can mix KRT25 successfully with SB6 and have an acceptable horse and an acceptable combination. But what happens if we mix SB6 and cook? We don't know. SB6 and sulfur, another unknown. But now look at this. KRT25 and curly unknown gene A, you will get bald patches. Okay. And cook and an unknown curly gene A, we don't know. SB6 and curly unknown gene A, we don't know. And KRT25 and curly unknown gene A can also possibly cause baldness. And one other thing that I want to emphasize on this, we have no idea what a homozygous curly unknown gene A horse looks like. My suspicion is it will be a bald horse, but that's only my suspicion. And these are examples. Now this is Domino. This is Veronica's horse here on the far left. And that is what is manifest. This is a summer picture of Domino's mother. And this is a picture on the right side of a horse that probably has some manifestation of curly unknown gene A that is very close to totally bald in the summer. I think that horse's name was Fancy, but don't quote me on that. And here's the family tree of this, and this is what happened. Carmela had curly unknown gene A, and she was heterozygous for it. And she was bred to a stallion that was heterozygous for KRT25. And by the little pundit square that I showed you at the beginning, we have a 1/4 chance of having a foal born with one copy of KRT25 and having a normal coat. We have a 1/4 chance of a foal being born with out a curly gene at all and having a normal coat. And that is Cora that you see here is the second horse, Curly Flash's Cora. And we have a 1/4 chance of a foal being born with one copy of the curly unknown gene A. And this horse in is just like her mother. And then sadly, we have a 1/4 chance of a horse being born with both. And that's what you get. That's Domino down there. And we need to avoid that if at all possible. That's why it is important to test your horses. This is a Zabaliskaya horse. This is telling me that we have something similar happening in their population. I don't know. We don't know a lot. War going on like it is. I don't think we're going to be getting anything more from Russia in my lifetime unless things change radically. But obviously, this horse has problems. Normally, just before winter, they shoot and kill and eat these horses in Siberia because they know that they can't survive the winter because they don't have heated barns and blankets. And so it's a hopeless thing to try to have that horse survive. So they normally kill it and eat it. And as we get towards the end here, not all curly horses are the same. We need to know that. We need to know what you're breeding. And you need to know, are the mutations that you're breeding compatible? That's the responsibility of every horse breeder or every horse owner that wants to breed. And this is kind of an interesting slide. I don't know whether that's an odd shedding pattern or something's going on at Saltwells Creek there. There may be another genetic something going on there. This particular horse was photographed by Pat Doak, and I just, oh boy, I don't know. It could be some unusual shedding pattern. I would feel better about this if I knew this horse was homozygous for KRT25, but being a wild horse, we don't know. But that's a little disturbing on that photograph. Now next, the importance, how do you know if you have curly unknown gene A? What you need to do is you need to test your horse for KRT25. If it is negative, if it has 2 copies of KRT25, then it's an extreme. If it comes up negative for KRT25 and has that unusual phenotype with that funny tail, then you know you probably have curly unknown gene A. And if that be the case, you need to not breed that horse to a horse that has KRT25 or Cook or SB6. You need to find a straight curly horse that doesn't have a curly gene to breed it to. That's the only responsible thing to do, because it is not fair to a horse to be born bald. And it's not fair to the owner to have to take care of it. Okay. So that's kind of a responsibility every horse owner has. And here's a case in point. I don't remember the name of this horse or the owner, but somebody posted this on Facebook saying, tell me about my horse. Okay. And this looks to me like curly unknown gene A, even though it's lost a little bit of the tuft at the bottom of the tail there. That looks like curly unknown gene A. And so my advice, and this is a gelding, so breeding isn't going to be an issue here, but if it was a mare that you were going to breed, the first thing I would say is test this horse for KRT25. And if it comes up negative for KRT25, then we know it is curly unknown gene A. And we need to go from that. Do not, if it was a breedable horse, do not breed it to another horse that has a curly coat, is what it amounts to. And this is a little slide at the end. And when this is posted on YouTube, These are some of the places that you can get your genetic data, genetics tested. You can get it tested through ICO, and ICO actually tests theirs either through A&M or from Etalon Diagnostics. The advantage of testing through ICO is it goes into the large Curly genetic database, and we use that as a basis for future research. And you can also go directly to Etalon Diagnostics. If you do that, it would be nice to send the results either to me or to ICO so that it can be kept in the database. And the same is true if you send it directly to Texas A&M, and they can, you'll get the results back. And it would be nice if you could send it on so that we can add it to our database. And lastly, UC Davis Veterinary Genetics Laboratory also does this work. And so those are several sources that you can use. And this is an example here of this horse here had Curly unknown gene A, the mare on the lower right. And she was bred to a quarter horse. And actually, we think that she was a dual gene with Curly unknown gene A and KRT25. And she was bred to a quarter horse, and the little baby only inherited KRT25 and is going to have a nice coat it can live the rest of its life, a normal life with, and not like poor Domino over here on the left screen that is dual gene for KRT25 and SP6. And that is the end of my slideshow.
Well, thank you so much, Dr. Mitch. That was a ton of amazing information. And I think it really fundamentally changes our understanding of Curlies as we've come to know them. Clapping on Zoom gets weird, so we won't ask people to unmute themselves, but a round of applause. Thank you for that presentation. Dr. Gus Cothran is up next. Dr. Gus is involved in research involving genetic mapping, genetics of specific characteristics such as coat color and hereditary disease, population genetics, and conservation genetics of rare equine breeds. Dr. Cothran was a four-time chair of the International Society of Animal Genetics, the Standing Committee for Thoroughbred DNA Typing Standardization, and a past chair of the Equine Standing Committee of ISAG. He is currently at Texas A&M University, College Station, Texas. And with that, I will let you take it away.
Well, thank you. I want to thank Mitch for giving me a great introduction. The reason I think that we can have productive interaction is that Mitch has all of these interests in the history of the horses, specifically that not kind of my background. And so he looks up all these things. And as he was going over some of those PCA plots, I was seeing things that he hadn't explained to me before. And I said, oh, that's why that's like that. And so that was always, it's very good for a scientist who's looking at the stuff like I'm about to show you to get an idea of what the horses themselves actually look like, because I don't see a lot of horses in my day-to-day. So what I'm going to talk about today is what we are trying to do to identify some of these other genes that Mitch gave us a good idea of what they're doing out there, but we don't know what they are. So, so I'm going to try to get started here on my slide presentation. We'll just have to see if this works. So, as Mitch explained, back a few years ago, we started looking for keratin, well, for a gene that caused curly hair in horses. And we found one that we thought looked really, really promising. It was the keratin 25 gene. You know, we got in our whole genome searching, we found a mutation that was in a keratin gene, and that made a lot of sense for a curly-haired mutation. But as Mitch said, when we started testing other horses, other than the ones that were in our panel, we were finding some that did not have keratin 25, but were curly. So we were working with the team in France at that time, and they came up with the keratin 25 just a little bit later than we did, but all of the curly horses they had were carrying the keratin 25 mutation. So they truly believed it, and we're not convinced by alternative evidence that that was the gene. And they looked up some other things, and it fit, the way the mutation affected the expression of the protein fit what we knew about the curly hair. And so we talked about it, and we went back, and about that time we found out from, I don't know if it was you, Mitch, or if it was Bunny or someone else, but somebody said, well, those horses that are not keratin 25, we call wavy. And so we realized we were working with two different genes. We also very early on realized that we had more than two genes because of the Mongolian horses and the Russian horses not having that gene and not being the wavy and such. So anyway, so what we're doing now is we're just going directly to whole genome sequencing. And these are the horses that we are trying to compare to each other and to control horses to see if we can find it. So we've got in the yellow up here, the Argentine curly horses. Then we've just got some curly horse samples, which I forget exactly what these were. I think that they're just kind of a random group of unknown curly horses, but as you can see, some of them they've all been tested for keratin 25 and the SP6, and some of them are missing mutations from either of those genes, whereas some of them have keratin 25. These are kind of controls. And then we have three horses that are Cook. So what we're looking at is trying to find differences in the genomes of each of these groups, and with the control group of non-curly horses. And we're going to find several things. So we've got in the yellow square finding of these US horses, we see 75 to 100% of the variants that we are finding are different from what we're seeing in the control group. And we've got a same group in the Argentine ones where we're seeing differences in variants. Mainly these are single nucleotide polymorphisms, or SNPs. And what we're looking for are SNPs with a frequency greater or equal to 75% in our target cohorts. And then we also are seeing some with very low frequency. So what we're looking for are differences in the control horses, the non-curly and the curly. And particularly, we're trying to see differences in the Argentine compared to the US and compared to the controls. And also the Cook horses are included in this. So we've got a good many variants identified, about 3,000 SNPs in comparison of the US curlies to the controls. Almost, well, over 4,000 SNPs in the Argentine population of horses. And then if you compare the US curlies and the Argentine curlies to the control, we only have two variants that are in common with the Argentine and the US. Now, with this next batch of slides, I've had a chance to go over them a little bit. These were put together. In fact, all of these slides were put together by the graduate student who is doing most of this work. Her name is Caitlin Castaneda, and she started working in my lab as a technician after having taken my genetics 320 class as an undergraduate. She is now a PhD student working for our colleague at A&M, Dr. Terry Rodsap. So anyway, she's doing all of this molecular sequencing and analysis. So Mitch provided all of these pictures. So again, the primary mutation associated with curly horses in the United States is that of keratin 25. However, as we've just gone over and seen from Mitch's presentation, not all curly horses have the keratin 25 mutation. And of course, we also then have the SP6 mutation, but not all curly horses, particularly those outside of North America, have that mutation either. So we want to see if we can find and identify any other genes that are associated with curly hair. Well, here's the family, the Swiss family that that Mitch talked about. We have sequenced all of these individuals, and we are looking for these to help us find another curly gene, and particularly the one that causes the balding. We don't really have any results to report on this at this point, although I will bring up something in just a little bit. So we have whole genome sequence from 21 curly coated horses. And these are the 13 from Argentina, 3 Cook horses, and 5 samples from the Swiss origin. And we also have then a large cohort. I think we've got, I may be saying the next slide, but I think we have about 400 sequences from non-curly horses. Now, one of the difficulties that we have in trying to do this analysis is, even with whole genome sequence, which is a tremendous amount of data, we only have 3 samples from the Cook lineage, and we only have 5 samples from the Swiss family. And this makes it very difficult to get statistical significance when you're comparing things that are in one group to things that are in another, particularly with the Cook horses, because we know all of these individuals are heterozygous. They only have 1 copy of the Cook gene. So we have identified sequence variants on all of these horses. The variants that are specific to curly coat populations had high frequencies in the curly population, close to 100%, but low frequency in the non-curly population, frequency of less than 5%. These are exactly what we are looking for. So we have some hope that some of these will actually mean something. I took out a bunch of slides that were showing some of the genes that we have identified variants in, but so far, with one exception, which I'll bring up in just a second, we've not found any variation in genes that we know are associated with hair structure. And we're also trying to look for things not only identified as being associated with hair, but we want mutations that could have a high impact on the gene product that they code for, possibly causing harmful mutations, but definitely changing the phenotype, or moderate impact, which they may change the phenotype, but possibly not have a severe effect. And then low impact, it's a harmless change in phenotype or no change in phenotype at all. And what we're doing is we're looking at the known function of all the genes that we're identifying with differences in curlies compared to non-curlies, and trying to see if any of the genes are fitting these patterns. And with almost 7,000 genes that we're looking at right now, it takes a little bit of time to get through this. And I should emphasize that this is something that Caitlin is doing on the side, and it's not directly related to her dissertation research. So initially we grouped all the curly horses together compared to the 400 control horses. We only found 9 high impact mutations. We found 91 medium or moderate impact variants, and low impact variants. In the curly population, there were 55. However, none of these variants were directly or known to be directly associated with hair structure. We're not sure, particularly Caitlin indicating that none of them were keratin genes or in the keratin gene family. We're not sure why we're seeing all of these. They probably are not things directly related to the curly horses, but are related to the ancestry of these curly horses as compared to the 400 plus control horses. So we're looking at now with each of these cohorts individually, the Swiss family, the US Cook horses, and the Argentine cohorts, and comparing them to the control. We're focusing on the Swiss family because it is the best defined subset of samples that we have, because we have father, mother, and offspring. The dam and baby and the baby 3 have curly coats, but do not have the keratin 25 mutation. What we have found now is 3 potential variants in the keratin 6 gene, which is responsible for structural integrity of hair filaments. So this is a potential candidate gene. At this point, we haven't finished the analysis of that so that we can say with any confidence whether that is actually true. And of course, this particular gene is the unknown curly gene A that's circulating in this family. And so this could also be the gene that is responsible for balding in some of these horses. Again, we don't know much about it except for what Mitch has just told you. It does seem to be an impact of 2 different genes and not so much just a single gene. That actually is something we have thought about as a possibility for quite some time. So anyway, we are at the point where we have identified a keratin 6 that may have a role in the balding of the horses. And Caitlin is continuing to work on this. She's going to be distracted a little bit for the next month or so because she is defending her dissertation 18th of April and finishing up. So she's in the process of writing up her dissertation now. She managed to get me these slides on very short notice. I talked to her about this, but she didn't quite understand that I was looking at March 27th as a date for needing these. So she got these to me Friday night. So anyway, this is where we are. We have a tremendous amount of data. Remember, each whole genome sequence is approximately 3 billion bits of information. So kind of burn up the computers in trying to do this. But anyway, we're beginning to find some promise. I think this family could be our breakthrough based on some of the things that Mitch showed on the principal coordinates analysis. We might have some clues on how to look at some of these things that could help us in identifying non-USA related curly mutations. And that's something I'm going to be talking to Caitlin about next week. So that's all I have. And I guess we can go for questions.
Well, thank you, Dr. Cothran, for sharing that with us. We appreciate it so much that you took time out of your weekend to share all that information. And yes, we will now go to questions. And I would just ask that people either raise their hand, which is something that if you hover over the bar at the bottom of the Zoom screen, there's a little thing that says reactions. And when you click on it, there's a button that says raise hand. And if you press that, I can see that your hand is up and I can call on you. And when you ask your question, obviously you will need to unmute yourself. That icon again is in the bottom lower corner of the screen. And also feel free to just share a thought or any feedback that you have. And if you do not feel brave enough to ask your question out loud, feel free to type it in the chat and I will read it for you. So I will turn it over to people for questions. Some more thank yous.
Surely somebody has some questions. Krista, give us a question about cook horses.
Okay, so if I would be prompted to breed Karma, is there a specific stallion that you would have in mind that would help specifically lock in the cook genes at this point?
Well, there are so few cook stallion horses out there. And the last time I looked, many of the stallions were getting elderly, you know, and I don't know how many are around anymore. So if there isn't a cook stallion available at this time, we might think about straight-haired curly stallion that has cook in the bloodlines, okay? And that would give you a 50-50 chance, according to the rules of Mendelian genetics, of passing that cook gene on to the offspring. And you'll have to get together with me about that, and I'll have to look into it to see what we can possibly come up with on that.
Okay.
When the time comes.
Let me just comment that one of the problems in looking at these cook horses and trying to identify a mutation is that they're all heterozygous. I know I mentioned that. But what you need to understand is that normally when you're looking for a gene and you're doing your gene mapping or whole genome searches and such, what you're looking for is something that the animal with the trait, all the animals with the trait have, and none of the animals without the trait have. Okay. So in this case, the cook curly is what we're talking about as the trait. But when they're heterozygous, you know, they've got one copy of this gene. It's probably the same in all of these individuals. I mean, there's little question that that's true. But the rest of the genetic background of these individuals may not be very uniform.
Okay.
And so it's just very hard to find. Remember, so we've got 9 billion bits of information to try to search through and find one of those that fits the pattern that we need. And that can be very hard. Sometimes, you know, sequencing can be a little bit tricky sometimes as well. You know, if the gene is in a good location, then there's no problem, but sometimes they can be in locations that are more difficult to get sequenced. And then the other problem is basically what we're normally looking for is a place where one of the bases in the chain of DNA bases is changed in the mutant animals compared to the non-mutant animals.
Okay.
But sometimes that's not what happened. What it could be that the gene that is causing the Cook pattern of Curly is actually a duplication or multiplication of a gene. That's very, very hard to find.
Okay.
In a very small, kind of quick survey like we're doing. So it's not always as straightforward as we'd like to hope it would be.
The other thing that I found extremely interesting was the overlap with the Mongolian, because what I found as I've moved different places, she began in Idaho where it was very, very cold, and her coat and tail and mane stayed full. Then when we moved to Illinois, where it's -40 in the winter and it's 100 in the summer, we saw a little bit more of the losing of the mane and tail. And now in South Carolina, I'm actually treating her more like a Swedish horse, even though there's not lesions or anything, but there is more of a loss in her mane and tail. So it's almost as though her genetics adapt to that environment very quickly. Whereas Sassy is a Damali Curly, and hers has remained kind of even keeled in her coat wherever we have.
What we're probably seeing there is something we call epigenetics, where it is control of gene expression by something that is not necessarily genetic.
Okay.
And is responding to the
environment. Okay. I found it interesting that in the Damali, my Damali horse not.
Yeah.
Dr. Mayfield, do you have any questions?
We
Can you
have
hear me?
about 4 questions on deck if you're
good,
ready for your
good.
next one. So Bunny's question was probably for Dr. Cothran. She asked, will it be possible to test for keratin 6 in other horses?
Oh, yes, absolutely. As soon as I don't know exactly what the variants that Caitlin has found are yet. When I saw this Friday night, that was the first I had heard about it. Okay, so, but absolutely unquestionable that once we know exactly what we have, then we can make a test that would be just like the kind of test we do with the keratin 25. And it'll be very, very easy to test for. And we will do that, obviously. And it may be that, we've got a Mongolian keratin 6 that's showing up in the Cook horses or something. Something like that is always a possibility.
So Dr. Wilkinson, I think the next question is to you.
Okay.
Tiffany says, so the alopecia or bald spots is not considered an extreme curly, but an expression of KRT and the unknown curly gene A.
Yes, that we can't prove that until we isolate the curly unknown gene A, which may very well be the keratin 6 that Dr. Cothran is discussing. Once we do that, we can go back and confirm what these horses have. And that way we have an exact match with that particular phenotype. So we can tell you definitively what would happen if these two got together. So, that's why it's so critical to find these genes and to test for them so that we can make recommendations to people on how to breed the horses, so we're not going to have horses that have to live a life with bald patches or no hair. Yes.
So we have a participant just labeled Hamrick. Do you want to go ahead and ask your question?
So my name is Deanna. I'm curious. I have the Mead appies, and you didn't have a whole lot to say about that. I don't know if you have more information on that. And just, I would be really curious to get together with you, Dr. Mitch, at another time and talk more on the breeding and the suspected curly type, and then have them tested, and then go back and compare that breeding because, well, one, I don't know a lot about the genetics of the curly genetics in the Appies or in the Meads specifically. But we do breed our Appy curlies to extreme without having her tested, not knowing for sure, but guessing it's the unknown A. And we've never gotten another extreme from them. It's just kind of, it's interesting. I find all this really interesting, and there's like so many variables. I don't know that there's really a question in there other than
Yeah, let me
talking to you more later about it.
Yeah, well, let me tell you about this. Probably in your breeding program, you have what I defined as an extreme, which is a homozygous or two copies of KRT25. And when you breed that to your Appies, you will not have balding in your horses, because the only possibilities are that you're going to have a homozygous, which would be possibly an extreme, or a heterozygous, which would not. And the LP gene, which is making your Appy horses, and possibly the pattern 1 gene if they have spots, or leopard pattern, that really won't have a lot to do with it. But I suspect from what you're telling me that you do not have curly unknown gene A in your breeding program. And that is why it is so important that we define these two things. And I want to say that an extreme is a homozygous KRT25, and the curly unknown gene A is something else. Okay, so that's why you don't have balding. Does that answer your question?
Yes, and maybe another time we'll get together and I'll message you and we'll chat some more about it.
Sure, be happy to anytime.
Curiosities. Thank you.
Yes.
So Marion, go ahead and ask your question.
Okay, sorry, I couldn't find my microphone. So I am a breeder, and my main foundation breeding stock used to be the Stackery horses, which had a lot of extremes, and I wanted to stay away from that. So now I have a combination with my Stackery stallion and a Cook mare and a Melly mare. Anyway, I get more straight-coated horses than curly horses. Is that something that I should pursue, or do I need to get myself a homozygous stallion?
Well, Marion, if you first you need to familiarize yourself with the Punnett squares and know what you're breeding. And remember, this is like going to Las Vegas and rolling the dice. If you have a 1 in 4 chance of winning that pot on the table and you roll the dice, it's a 1 in 4 chance. And so everything is
Also
calculated
remember
with
that
odds. What's that?
Also remember that it's always a 1 in 4 chance. That's why, even though
Yes.
it's a 50-50 chance of getting a boy or a girl with your child, you can still get 6 girls in a row because it doesn't always. This is a statistical average. The 1-2-1. That's a statistical average. It's not every time.
Yeah, so I'm
It
good.
is important to understand that.
So, like Gus said, Marion, you may be unfortunate in getting 6 girls in a row here.
No,
it's just okay, and it's just the odds of the statistical odds of that particular thing happening. Now you can increase your odds. For instance, if you had a homozygous KRT25 stallion, then you would every foal would end up having at least one copy of KRT25 and be curly. But depending on who you bred that horse to, you might have a 50-50 chance of having an extreme, which would be a homozygous KRT25. If you a homozygous stallion who is bred to a straight mare, that's why I say straight curly horses are worth their weight in gold. If you had a homozygous stallion and a straight curly mare, you're guaranteed 100% of having a heterozygous curly, which is the most attractive type of KRT25 every time. It would be 100% probability that you would that every foal would have that. And that way you would have a very marketable horse.
Yeah. So then I recently got a pony from Canada, and he is tested, and he has the KRT25 and the SP6 gene.
Okay. That's a more complicated Punnett square, giving you more odds. And okay, so in that case, you can have, depending on the mare you bred it to, you could have another dual gene horse like the father. You could have a horse that only inherits KRT25. You could have a horse that only inherits SP6. And there's a possibility that you could have a horse that doesn't inherit a curly gene at all. Okay, and
Do I need
so
to have
that's
the mare test?
What's that?
I need to know, but I do need to know if I have a unknown curly of the type A curly in my mares. Then.
Look for that telltale phenotype on the tail. That's kind of a clue. And if that shows up and they test negative for KRT25, there's a good chance that's what you have.
And do you know if the Farrell horses, or I don't know how you pronounce it, the Frostfire horses, have you tested that line?
You know, I'm not familiar with that line, Marion. So you'd have to give me some more info, and I try to look it up for you.
Yeah, because okay, I will send you some information because they all have that bushy tail.
Right. Yep.
Okay, I will get in contact with you about that.
So Marion, just to clarify, was that the Friedel line that you were talking about?
Yes, that was.
Okay.
Yeah.
Oh, okay. Now I'm familiar with it. I'm sorry. Yes. Okay.
Yeah.
I don't know if that changes your answer, Dr. Mitch, but
well, no, they were heavily bred into quarter horses, and Dr. Cawthron and I are looking into that. I wanted to see how close those particular horses are to quarter horses, and that'll give us a little thing. But as far as curly unknown gene A, that goes all the way back to the early Demelli and Mead breeding programs. So it's like it pops up all over the place because it goes back to the beginning of curlies in domestic curlies.
So one specific I have one question that everybody always asks and where everybody's always fighting about.
Yeah.
Is the curly horse a breed or not?
Well, and I'm going to let Dr. Cawthron answer this also, but to me, I'm working on trying to resolve that issue, and curly horses in general, because they are so many different types of horse and dealing with so many different types of mutations, don't really fit the definition of a breed. And obviously, a Zabeletskaya horse in Siberia is a different horse than a Demelli horse. And so curly horses in general don't fit the criteria of a breed. But some curly horses may come very close and actually fit the definition of a breed. And
Like the Russian
but
one.
yeah, but you have to take separate groups of them out of the whole. Does that sound right to you, Gus?
Yeah, I
Right,
mean,
yeah.
the problem with calling curlies a breed is that it's pretty much based upon a single gene. Okay. And that's, as we just talked about here, you know, if it's a stallion that's only heterozygous or something, or a heterozygous horse breeding, only 50% of the offspring will carry that characteristic. And so the key thing for a breed is that the characteristics are found in every individual of the population, and that they pass those characteristics on to the next generation. Now, in the United States, we don't have any rules for what is a breed. So if you get together and form an organization and call your horse a breed, as far as the law is concerned, it's a breed. In Europe, it doesn't work that way, or Canada even. So it's variable, it's a little tricky, but the biological definition basically is based on overall physical characteristics, and that all members of a breed should have the same ones, and that they're heritable.
Yep. Okay. Thank you very much.
So the next question is from Melanie. She asks, wouldn't it be easier to look at gene expression for identifying the unknown gene or the differences between the phenotypes?
I'm going to let Gus answer that one.
Yeah, I thought so. I'm not exactly sure what you mean by that. Gene, you can't look at gene expression if you don't know what the gene is. Now, we can look at the hair characteristics and things like that, might give us insight. However, in the past, before we found the keratin 25, there was nothing that we had seen that I recall. Maybe you have a different memory on that, Mitch, but I don't remember knowing anything from the structure of the hair or the follicles or things that told us to look for keratin 25. There's so many keratin genes that's difficult to start with. So it's really kind of a hard place to start, actually.
Hopefully that answers that question. Dov Cardenas has a question. He says, should we still avoid breeding a sulfur curly, AKA renegade, to other curly horses now that more is known about balding / unknown curly gene A?
Yes, let me answer that one. Absolutely, Dov. I think that we need to keep the renegade's unique curly mutation separate and not mix that with other curly mutations. We have no idea of what the implications of that would be, and we have so few of the sulfur horses. Maybe someday when we have more feet on the ground and that particular mutation is more common, we can actually test and try to isolate it, find out what it is, and possibly what gene, what chromosome it's on, what gene it's in, and go from there. But for right now, we have, we really and truly, we only have Renegade and his sperm, and that's all that's keeping us from extinction. So I would really hate to see him bred to another type of curly horse. Does that answer your question, Doug?
I might say that it wouldn't hurt to breed him to straight-haired horses and try to get the mutation more plentiful in the horse population.
Absolutely. I think breeding Renegade to a sulfur horse would be a great advent. Currently, we are starting a Spanish sport horse curly breeding program with Renegade, where we're breeding Renegade to Lusitanos and Andalusians, and trying to retain that curly gene. And one of the problems with sulfurs is that they're not very large in stature, and that limits the market value of them. And so I'm basically here trying to follow the model of Curly Jim, where we started out with a single horse and managed to breed in Missouri Foxtrotters and ended up with a really wonderful line of curly horses. And I'm hoping we can do that. Somewhere down the line, I would like to see more pure sulfur type horses. And remember, Renegade is half Wilbur Crusay and half sulfur. And even though they are closely related, again, he's not quite pure sulfur, but he does carry that gene, and we're trying to keep that gene alive. Yeah. Does
So
that,
the next question is from Tiffany.
yeah?
He says, how do you preserve the Salt Wells type curly if the BLM tends to geld most studs before adoption to the public?
It'll have to be that second generation. Those mares will have to be bred, and actually, if you have an exceptional horse and you contact the BLM, sometimes they won't gild one, and especially if it's young. So, for instance, if you adopt a 6-month-old curly coat from Salt Wells Creek, and you ask the BLM not to gild that little foal, that little colt, they normally won't do it. And so you can then adopt that horse and train it while it's young and have a breedable stallion from there. But an older horse, they will almost always gild.
And Dr. Mitch, just a follow-up question to that. Salt Wells is KRT 25 and Fish Creek is also?
Yes, although Fish Creek may have Cook in it. It may, at one point, that curly gene A in it. The Fish Creek is kind of special. Now, Fish Creek has been picked over and gathered so many times over the years that I'm not sure how many genes are available still in the population, but by far, the most common gene found at Fish Creek is KRT 25. And the Salt Wells HMA was probably seeded by curlies from northern Nevada. There was a Laramie stud and Rocket that were taken up there and let go into those herds. And even though their conformation with simply one stallion trying to breed up there didn't really alter the conformation of the Salt Wells Creek Mustangs, they did seed that with a dominant mutation, which has carried on through the years. And I think those horses were taken up there in the 40s or the 50s, but I'm not sure about those dates. Okay.
Krista, would you like to ask your question?
Yes, ma'am. I noticed that with the unknown A, the very distinctive tail ending, Karma, the first couple of times, the first 2 years down here, she shed out. The dock did not become that bald, but we had the floof at the end. But her tendrils are extremely thick. Is that the same with the unknown?
No, it's the unknown has extremely short hair on the dock of the tail. You know, where Karma and some of the KRT 25 horses, my old mare Shelby had a tail somewhat like that, but it was kind of thick, curly hair, more like a body hair on the dock of the tail, and then it kind of fluffed out. And my daughter kept saying, Oh, that's, your horse has a tail like they use on pin the tail on the donkey.
Donkey.
And, and it's kind of, again, that's kind of an unusual tail morphology, but it's
Yeah.
not like that unknown gene where it's just practically naked winter and summer on the dock of the tail, and then you've got that puff at the end. To me, that's real characteristic of that phenotype of that particular gene. And that the mane is never gets very long. It's like really, really short, if you notice on those horses.
Okay.
So those are the two things that stand out to me on that.
All right.
And then of course that curly unknown gene A, you know, like Karma doesn't get bald in the summer. And
No.
you can see how it's not really a true baldness, but they get really sparse on coat in the summer.
Right, right. And another aspect that I've noticed is where the other girls are like meek and mild, like if they're at the hay hut or whatever, there's no tail flipping. The least little bit of heat and Karma's tail starts going up and down in a much more aggravated way than like she's fly swapping even if there's nothing out. So I just kind of wondered that.
Yeah, maybe she doesn't have as good a fly swatter as the other horses. I mean, I don't know.
We're working on it.
Yeah.
Thanks, Doc.
Yeah.
Marion, go ahead and ask your question.
No, I accidentally hit the wrong button. Sorry.
Okay, that's totally fine. Dr. Mitch, I have a question for you. For all of the rare curly genetics that are endangered, is there a preservation effort ongoing for each of those?
The only preservation effort that I know is me. Okay. And I don't know how many people listen to me on that. You know, I try, but I may be kind of the voice in the wilderness there, screaming and trying to get everybody to listen. But hopefully, by my squeaking a lot, people will put some grease on that wheel. And I'm not capable of saving all these horses. You know, we have to do this as a group of curly owners. And maybe Joan has something to say on that, you know, because she's head of one of the registries, and maybe she could make a comment about anything involving that.
Feel free to jump in, Joan. Just make sure you're unmuted, lower left.
Hi, can you hear me okay? Yeah.
Yeah.
And what specifically, Mitch, did you want me to speak about?
Oh, the
Mention-
preservation efforts of the rare types of curlies, which I know we're involved with the Renegade thing, trying to do that. But
Right.
I don't know that there's many people involved trying to preserve the Cook line of curly horses, you know, and
Yeah, I think on some of the lines there are individual breeders, but definitely not all of them.
yeah. Yeah.
Yeah.
Is there anything we could do as an organization to help on that? You know, it's because it's kind of up to a group of individuals, and since there's so few of them, you're probably down to 4 or 5 individuals that would have to do something at this point.
Right. I think that we need to do more of what you and Gus are doing today. It just,
Yeah.
you know, education needs to be heightened about this. So.
Right.
Yep, we need to keep publishing this information and disseminating the information. And yeah, it's something that takes a group effort. And you know how the cogs of a wheel are just so few sometimes to make something go around. We need to stick together and try to help each other do this.
Right. Right.
So just focusing on like the Renegade idea, it almost takes all my brain energy just to focus on one asset, you know, one direction. So
Right.
it's really nice. Like Krista with Karma, I mean, that kind of got me excited when I heard your comment, you know, what should I breed Karma to? Because I was thinking that earlier when this started, and I was thinking, what would be a good stallion to breed Karma to? Because I don't think Krista's ever bred her before. And boom, you came up with the question. And so that, I'd love to be part of the think tank to, of thinking about horses that would be appropriate for Karma to breed through. That would be fun to do that. And you mentioned about it being a cook gene, a cook stallion, but my mind goes to what if it was a straight coat, non-curly stallion at all? You're looking at a 50-50 chance of that gene being passed through. I don't know. I think there's some value in that as well. But Dr. Cothran and Wilkinson know the best.
Oh, no,
Yeah.
I don't. We
Yeah.
need all the help we
No.
can get.
Well, and I'm not a breeder. I'm a happy home.
Yeah.
So it's kind of it's a scary thing to think about breeding one of your babies and like
Right.
something going wrong, or and you want the best aspect for them. And along the lines, like I said, I've been thinking a long time, what is a good way to try to preserve this particular line? And honestly, breeding is always at the top, because you can't really harvest anything else that's going to propagate in that same manner. So guidance would be, and I am open to it, as long as I had guidance, you guys.
Yeah. And I think probably the most disastrous route would go to breed to a curly stallion who is SP6 or something, because you don't know what you're getting. I guess you could test the foal.
Exactly.
But if there's anything else in there mixing, you wouldn't know what's being expressed.
Exactly.
Yet. Yeah.
And she's such a smart girl, and she's such she wants to please, and she's opinionated, but like she has a curly eyelash, and she leans over and allows me to snip the curly eyelash to keep it out. So, there's so many traits in her I would love to see passed forward. I just want to do it where the foal would be comfortable with things. So, and something, and there's always odds something will go wrong, but to listen to this aspect of things and to think that her line would just end, that's scary too,
Yeah.
and to a point
Chris,
that
I just saw a little blurb on the bottom of the screen here from Bunny. And I think Bunny would be instrumental in this, because she knows these pedigrees like the back of her hand.
Yeah.
And has the whole database in front of her. And she just sent a little thing that there is a cook stallion in Germany. And and so I think a place to start with this would be to start with Bunny and have her look through the genetic things for aliens that we know about, and see what is available, and make that one of our options. Okay. But I think the other option is what Joan said, there, to try to find a suitable stallion that isn't carrying a curly gene that we can breed and have a 50-50 chance of having a cook come out and not mix the other curly genes with it. Yeah.
Yes, sir.
Yeah, and just email me and I'll try to help. And we'll get Joan and Bunny and myself on board on that, and we'll look into it.
All right. I appreciate you guys. Thank
Yeah.
you.
And Bunny, if there was anything else that you wanted to say about that, please feel free to jump in. And also, if you have the link handy for the ICHO genetic testing page, if you would put it in the chat box, that might be useful for people as well. And maybe Bunny isn't going to jump in. Marion asked about harvesting eggs with rare genetics with the situation with Krista's horse.
That's a possibility, but it's terribly expensive. It's fair. I don't know the exact cost, and we could look into that. That's another possibility, and have we can see about that. The best thing that we can have to preserve the cook line is just what we're trying to do with Renegade, is have a stallion that we can, that's trained to the dummy, that we can start shipping sperm around and get, it's basically a matter of getting more feet on the ground is what it really is. Would you agree with that, Gus?
Absolutely. The copies of genes is what your goal is.
Right. Yeah.
Marion also commented that there is a warrior line preservation group. And Marion, I see you have your hand up, so feel free to jump in.
Yes, there is a group of women who are very particular about the warrior lines, and they go a little far in my opinion, because they say that that's the only best line in curlies there is. But just to throw it out there, there is a group of ladies out there who try to preserve the warrior line. Yeah. So, but coming back on this Cook line, so I do have a daughter of Houdini, who unfortunately tested negative for the Cook gene.
No,
But
no,
if we
Marion,
find this,
we can't test for the Cook gene. We can't test for that gene yet.
We don't know what it is yet.
Yeah.
oh, well, okay, so we tested for Houdini, so that because she was out of Houdini.
Yeah. So that means that, is she
So-
curly? Is she curly?
Yes, yes.
Has
Well,
she
you
been
tested
tested?
her. We
Has she
said
been tested for KRT25?
negative.
Oh, so
Yes.
she's negative for
Yes.
KRT25? Okay. And she's curly?
No,
Oh, she's not curly.
yes. She's not, she is curly, but but I thought you wanted to know at that point that she was a Cook horse or not.
Right, right. Now, hold on, Marion.
So Bunny is posting,
know
she has KRT 25-1 copy. Bunny doesn't have a
Yes,
microphone.
Oh, okay, okay. She has, and
But that's correct.
Right.
That's correct.
so there is
But my question,
a slight possibility she
Go ahead.
could be dual gene and have the Cook and KRT25 at the same time, but we don't know that because we can't test for it.
oh, okay.
We don't
So,
know what
but
it would
my
look like.
question would,
And right
So,
now,
my-
Marion, Gus and I are starting to begin some work on the warrior line of horse and come up with some conclusions on those. And keep in mind that the warrior line of horses was heavily bred with quarter horses at the beginning,
yes,
almost like Missouri Foxtrotters were to the curly gym line. But how
Yeah,
close they
I'm-
are to a quarter horse, I don't know yet. We haven't run them.
very close. The way mine looked, very close.
Yeah, and that may be. Yeah.
Yes, yes. I'm not a fan, but that's another. But okay.
So Deanna had a question saying, by more boots on the ground, do you mean more genetic testing for our curly horses?
No, I mean, more living individuals that carry these rare curly genes is what I mean. When we're down to just 3 or 4 or even 1 in the case of the sulfur, it's critical that we get more living horses that carry those genes, or else they are just going to go extinct.
I hope that this webinar helps the entire community come together. And so hopefully we can make more efforts to keep that from happening. We can give you a hand, Dr. Mitch, in saving all these horses.
And let me make one last little statement here for
Sure.
ending this. And I'm going to go back to my beginning statement, which says, For too long, the horses have been the subject of speculations, legends, and total fantasies, which are not based on any empirical evidence whatsoever. This has led to rivalries, misunderstandings, and disputes about philosophies, which are totally based on fantastical speculations, much to the detriment of the horses involved. And we need to all remember, this is not about your ego or my ego. This is about the horses. It's also about future generations and preserving something worth preserving. It's a cause which is far greater than ourselves. And all of us need to put our differences aside and do the right thing for these horses. And that's the bottom line of everything.
Thank you, Dr. Mitch. That's very well said, and I couldn't agree more. Before we wrap things up, would you be willing to talk about Curly Horse Genetics Part 2, when that's happening, and what you plan on addressing there?
Yes, I'm going to try to recruit Gus on this webinar also, that coming up in a month or so, and if he's available. And what I wanted to do is I wanted to talk about the different types of mechanisms of the mutations, what a SNP is, go into greater detail about how to do Punnett squares for breeders so that they can understand the odds of what they're doing when they're breeding. And especially since there are so many people out there now that have dual gene horses that have keratin-25 and SB6. And I also wanted to explode some of the old original legends of curly horses, like curly horses came across the Bering land bridge in Beringia and somehow filtered down to Nevada. I wanted to, explode some of those old myths, which actually are hanging on and actually hurting us. And the idea that all curly horses are the same, just a whole bunch of things like this. And so that's going to be the subject of the second one. And the third one after that is going to be how can we set up these registries to preserve curly horses and have a meaningful registry with all these diverse types of horses. So that'll be the third one. Okay.
And we're planning on doing Part 2 on May 1st, which is a Sunday. And it'll
Okay.
be at the same time, 1 p.m. Central Standard Time. Correct?
Sounds good. Yes.
So Bunny posted that the ICHO is now adding seals to registration certificates to show what gene or genes the horse has if they've been tested, which is an exciting development. That's great to hear about. Thank you, Bunny, for sharing that. And just before we wrap up and I end the recording and end the meeting, I just wanted to say thank you so much to Dr. Mitch and Dr. Gus and to Diane for putting this together. And thank you to everyone who participated. It's really lovely to see so many people here and have so many important voices all in one room. I'm so grateful to be part of this. And last but not least, Dr. Mitch and Dr. Gus, if you could just talk about ways to get in touch with you. You know, if people wanted to private message me on Facebook, I'm happy to pass questions along. But if people are wanting to preserve some of these different genetic mutations, how would they go about contacting you? And Dr. Gus, why don't you answer first?
Well, the best way is through email. You could post by email to the people. That would be fine. I never can remember exactly what the link is, but the lab at Texas A&M has all the addresses and things. So I don't seem to be hard to find.
And Dr. Mitch, how about you? Can people contact you through Messenger?
The same way. I can email would probably be the best. People are always welcome to call. It'll probably have to be on the landline if they call. But if you were to talk to Diane Mitchell or Joan Henning or Bunny or any of the people in any of the registries, they would be happy. I give them my permission to give somebody my email, and then we can make some contacts. I think most of the people that I've seen that participated today probably already have my email somewhere, email address. And it's wdocmitch16@yahoo.com. And if you can, but you can certainly get them from them. And I am always happy to hear from Curly people. So just drop me an email if you need to.
And Bunny, if you're still here, would you mind typing? You handle some of the inquiries for genetic testing, correct? I'll just let you respond in the chat about that. Bunny says yes, she does. So Bunny Ravaglia over on Facebook, and also if you call ICHO, I believe, can help you figure out genetic testing. So I think that's it for questions today. Bunny says email is best. I want to just say thank you to everyone one more time. And we hope to see you back here May 1st at 1 p.m. Central Standard Time. Thanks so much, everyone.